Hi guys, back again with the next chapter for Biology 242, and this one concerns blood. So I already made a video for the lab portion of this, but of course we're moving on to lecture, since it took approximately 8 million years to record all those reproductive videos. So as much as I think reproduction is fascinating, and I'd love to talk about it all day, I'm excited to be switching over to blood, because it's also fascinating, and it's our launching point for both the circulatory system in general and also the immune system, with the immune system being especially topical at the moment, given that we are living in the time of a pandemic. So, with that, away we go. So, in general, with regard to functions of blood, why have it? One of the reasons is that diffusion is too slow. Here's what I mean. So imagine that a molecule of oxygen needed to diffuse from my heart down to my foot because I have, metabolically active tissues in my feet. The circulatory system allows that to happen super rapidly because blood containing oxygen is being actively pushed from tubes by my lungs to tubes in my feet. Perfect. If I did not have a circulatory system, that diffusion would take approximately 27 years for one molecule of oxygen to get from my heart to my foot. It's a long time. So we don't have time to wait for diffusion, is the take-home message there. We have to speed diffusion along by using a circulatory system to physically move things past each other. So one of the things that moving things past each other accomplishes is it increases the steepness of a concentration gradient and keeps the concentration gradient high. And that is another reason to have blood, in addition to transportation and distribution, regulation of interstitial fluid, all of the interstitial fluid in you, so the fluid that's between your cells and tissues, all of that stuff comes out of your blood. So the pH of your blood is going to influence the pH of your interstitial fluid as well as what ions are in it. And we know that cations are really important for regulation of things like nervous and muscle functions, so let's add some of those. So we've got calcium, that's a big one, but also sodium and potassium. Those are kind of the big three. If you get significant dysregulation of any of those ions in the interstitial fluid, bad stuff starts to happen, and we've talked about that. Changing blood volume can help with restriction of fluid losses. So we talked about atrial natriuretic peptide and also the renin-angiotensin system. Defense is a role of the immune system. And thermoregulation just means that blood is mostly water, and water gains and loses heat slowly. So if you change where blood is in your body, you're going to necessarily change how warm or cold a part of your body is. So this is why, for example, hands and feet might get cold if the ambient temperature is below your body temperature. That's because blood is being moved away from your extremities and towards your core to help keep you warm. So as far as blood, and this is whole blood, so if you just stuck a needle in me and took some blood, didn't do anything to it. Blood is about 5 times as viscous as water, meaning viscosity is resistance to flow. So if you're not familiar with viscosity, think about maple syrup versus water. Maple syrup is the more viscous of those two fluids. So blood is sticky and cohesive, and this is due to both dissolved proteins, of which there are many in the plasma, and also formed elements, so cells. Cells and also parts of cells. So let's put cells and cell bits. So the pH of your blood should be just north of neutral, so slightly alkaline. And when we get to the respiratory system, we'll talk about how breathing influences this pH. Depending on how big you are, you may have anywhere between 4 and 6 liters of blood. And this brings us to an interesting but not testable estimation. So this would be a necessary skill for you if you are an emergency medical technician, or if you, for example, work in the trauma department of an emergency medical services unit. So, for example, a 100-pound person has about 3.5 liters of blood. A 9-pound infant has about 0.28 liters of blood. So if you multiply in general 0.07 times weight in kilogram, you can get the liters of blood that's in a person. So if this is your patient and they're losing blood, you can estimate how much blood they had to begin with and then relate that to how much blood they would, basically how much blood they could lose before there was a severe risk of, say, hypovolemic shock. Very talented emergency medical staff can estimate blood loss by looking at things like absorptive pads, trauma pads that are put under patients, and actually make on-the-fly calls about transfusions based on those things. So if you're curious about that, you can read the accounts of, for example, Army medics and such who describe making that calculation on the fly. It's an impressive skill, but you only get to use that skill when things are very, very bad. So components of blood include plasma, which is mostly water, dissolved ions, and proteins. The formed elements are primarily red blood cells, less than 1%, less than 0.1% white blood cells, and less than 0.1% platelets. So the white blood cells are really, really, really tiny. So let's talk about plasma first. It's about 55% of whole blood. That's going to vary a little bit with something called hematocrit. So hematocrit is the amount of your blood that is made up by red blood cells, and so that is going to be related to plasma because it's a percentage. So if you change the percentage of plasma, you're going to change hematocrit and vice versa because those numbers have to shift to equal 100, right? So this has a range, obviously, also depending on hematocrit, of between 46 and 63%-ish on average. Plasma is mostly water, but there are also plasma proteins. And these include albumins, which are a group of small proteins made by the liver, but they contribute strongly to what's called osmotic pressure. Globulins, these are antibodies, among other things, but primarily antibodies. So these are proteins that stick to pathogens. And fibrinogen, which is a precursor for fibrin, which is responsible for clotting. There's also solutes, including electrolytes, like the big three cations I mentioned, as well as nutrients, because of course, the blood is how nutrients get delivered. Wastes are also carried away from tissues by plasma. So they don't enter the red blood cells in general. They stick around in the plasma. So these include urea, uric acid, creatinine, and bilirubin. So this is coming from skeletal muscle. And bilirubin is a breakdown product of hemoglobin. It's secreted in bile. Respiratory gases are, of course, also carried in the blood. So those are going to be oxygen, molecular oxygen, that's O2, and carbon dioxide as well. So plasma proteins are produced by the liver, by plasma cells, and by endocrine organs. But the liver is the primary mover and shaker of this particular corner of physiology. So what that means is liver disease, such as cirrhosis due to chronic alcoholism or cancer of the liver, can change the liver's ability to produce plasma proteins and cause dysregulation of them. And what that ends up doing is changing the viscosity of the blood and also changing colloid osmotic pressure, which is the ability of blood to attract water. So changes in plasma proteins end up having pretty large consequences on the characteristics of blood, which means that your liver is related. So this is the link between your digestive system and your circulatory system. So plasma proteins remain in vessels, which just is another way of saying plasma does leak out of your continuous capillaries with pressure in the capillary bed. That forms the interstitial fluid, but the plasma proteins are supposed to stay in the plasma. They're supposed to be too big to get out. If you end up seeing plasma proteins escaping, that's indicative of some kind of compromise in capillary integrity, which of course is not ideal. So albumins as a group maintain the colloid osmotic pressure of the blood, and they're also little sticky taxis for various hormones. So fatty acids, which are of course insoluble in water, as well as thyroid hormones and steroid hormones, are transported, some of them, on albumins. Globulins are about 35%, and these are primarily antibodies. There are other globulins that exist, but we're not really going to discuss them. So think of them as being immunoglobulins or antibodies. The ones that are not immunoglobulins tend to have transport roles, but we're not really going to discuss the specifics of these. One of them you already know about, and that is thyroglobulin and transthyretin. But there are others. Fibrinogen is a precursor to fibrin, that is for blood clotting. So fibrinogen is soluble, meaning that it stays in solution. Fibrin, its outcome is not. So when you get the conversion of fibrinogen to fibrin, the fibrin precipitates out of solution and begins clotting. And then the remaining 1% are things like angiotensinogen, peptide hormones, and other things. Moving on to the formed elements, we've got platelets. And platelets are not actually cells. So they're called thrombocytes in some literature, implying that they're a cell, but really they're a fragment of a cell. So platelets are actually small fragments of a large cell called a megakaryocyte. Megakaryocytes live in the bone marrow, and they basically extend little pseudopods into sinusoids in the marrow spaces, and the blood flow in those sinusoids breaks off small chunks of the foot of the megakaryocyte, and those little chunks become platelets. Leukocytes are white blood cells, and these are less than 0.1% of whole blood. Erythrocytes are the majority, making up 99.9% of the formed elements. Excuse me. Paroxysmal yawn there. So erythrocytes are sort of the workhorse of the blood, and they are also way and far away the most numerous. So this picture that you've been seeing for the past few slides is a picture. It's a false color, meaning that although the blood cells are really red, you wouldn't be able to see this on this microscope image unless someone had colored it. So this is a false color scanning electron micrograph, which is a kind of microscopy that you can use to resolve very, very small things. Because these are only about 7 microns in diameter. They're real little. Red blood cells are constantly produced and are constantly dying because they don't have a nucleus. More on that later. And so that's because of not having a nucleus and not being able to do things like replace proteins, etc., they have a limited lifespan. They only live about 120 days. During that time, however, by being pumped around through the vasculature, they travel around 700 miles, which is impressive. So that's basically their sort of shelf life, so to speak. You have between 4.5 and 5.5 million of these per microliter. So one drop is about 50 microliters if you took like a dropper of blood. So if you've used a pipette, for example, if you've taken micro, I know some of you have, you know how tiny a microliter is. It doesn't even look like the pipette tip is full. It's so wee, and yet there's millions and millions and millions of red blood cells in each of those little tiny droplets. And that means that there's about 25 trillion red blood cells per body, give or take. Trillion with a T, not billion, trillion. That's incredible. Okay, so hematocrit I mentioned previously. This is the percent of blood volume. That's what BV means here, blood volume. The percent of blood volume occupied by red blood cells. And there's a little bit of sexual dimorphism in this, primarily because of androgens and the role of androgens in stimulation of mitosis. So on average, folks who produce a lot of androgens end up having a little bit higher of a hematocrit. But of course, those are averages across huge sample sizes of humans, and so individual results may vary. So specifically, androgens affect erythropoietin, which is that kidney hormone. And the kidney hormone, of course, acts on bone marrow. I know it looks like it says bone, but it says bone. So a hematocrit tube is a tube that is put into a centrifuge to estimate hematocrit. You spin down the blood and you estimate based on tick marks on the tube what the hematocrit is. So here we have 100% of blood, the buffy coat, which is the little divider of white blood cells and platelets in the middle, and then the packed cell volume, which is what PCV stands for, is a stand-in for the hematocrit. So with regard to RBC structure, red blood cells are biconcave discs, meaning that they look like an almost donut. So a torus is a donut. A toroid is an almost donut. So toroids look like basically flattened frisbees, where they have like a thin spot in the middle. And red blood cells are shaped like this for a reason. One of them is so they can flow through tiny capillaries in a stack, and the other is so that they can fold into a little taco shape when they have to pass through the smallest blood vessels. So if they were spheres, they could not fold. So as I mentioned, they have about 120-day lifespan. And this is from start to finish. So a small number of circulating red blood cells are called reticulocytes. Here, I'll write that word down. And these are red blood cells that are finishing up forming while they're already circulating. So they're called reticulocytes because they have this sort of lacy network of degenerating organelles inside of them that they will discard and then achieve their final form, which is this little frisbee-like guy. So basically they become a sack of hemoglobin. So 95% of the protein in the cell is hemoglobin. That's basically all they're for. So to help you understand why and how they don't have a nucleus and don't have organelles, they also don't have mitochondria, imagine the following. So I'm going to move on to the next slide in the meantime. So I'm going to tell you a story while I navigate here. So before I talk about hemoglobin, let's examine the reason for the phenomenon of red blood cells not having much in them except for hemoglobin. So to do this, I'm going to tell you a story, and in this story, I am an ice cream man. So it's COVID-19, and maybe I'm out of a job, and maybe there is a demand for mobile ice cream to bring joy to the people, and so I take this job as an ice cream person. So when I get the ice cream truck, it's full of ice cream, and my job is to deliver ice cream to people who want ice cream. Here's what my job isn't. My job is not to eat all the ice cream on my way to the people. If I did that, I would be literally the worst ice cream man in the history of ice cream men. I would just be gobbling up the ice cream on my way to the children and then get there and be like, oh, sorry, kids. I ate all the ice cream when I was driving here. Goodbye. Good day. So the reason, sorry if you just heard that flapping, my bulldog is trying to get my attention. The reason that there aren't any mitochondria or anything requiring ATP really inside of red blood cells is so that they don't gobble up the oxygen on the way to the cells that they're trying to supply. And that's really important. I do want to stress that that's not the strategy for every vertebrate. So, for example, birds and amphibians and fish, they tend to have nucleated red blood cells, which means that their red blood cells are capable of much longer lifespans than ours are. So if we experience any interruption in red blood cell production, that's what's called aplastic anemia, which is the anemia that results from the lack of making new ones to replace the ones that are dead. So hemoglobin itself is a tetramer. So this is a word that you may remember from Biology 160. Tetra means 4, and mer means there are 4 protein subunits that come together Voltron style to form the total protein. So we have in our hemoglobins 2 alpha globins and 2 beta globins, making 4. And in each of those globins is embedded something called a heme group. A heme group is a pigment molecule. Full disclosure, most pigment molecules are shaped like this, so they have multiple carbonaceous rings, and that structure causes them to reflect certain wavelengths of light. So this pigment complex has a central ferrous iron in it. Ferrous iron is Fe2+. And this is what the oxygen molecule is grabbed onto with. So oxygen has a high affinity for iron. If you've ever looked at rust, you know that. So like rust on a metal is oxidation of metal. Oxygen loves to gradually destroy iron. That's what rust is. And that's also the reason why blood smells like pennies. Surprise! So each heme group binds with one oxygen, meaning that a hemoglobin can carry a total of 4 O2 or 8 oxygen atoms. I'm reminding you of this specifically because I want to remind you of the principle that molecules, which are atoms covalently bound to each other, are different from atoms. So if you have 280 million hemoglobin molecules in each red blood cell, and then you multiply that by 4, that means that one red blood cell carries over 1 billion oxygen molecules, which is crazy. So it's very, very efficient as far as carrying. And as long as you're not being a bad ice cream man about it, it means you're efficient at dropping stuff off too. So hemoglobin binds reversibly to oxygen via that iron. So the place on the hemoglobin that oxygen binds to is primarily the iron ion. So when hemoglobin is bound to oxygen, we call it oxyhemoglobin. Interestingly, hemoglobin also transports the waste, which is CO2. However, hemoglobin does not bind CO2 at the heme group. CO2 kind of just hops on here. So maybe it's like, I'm going to sit over here, for example. Or maybe there's one over here. And that's because CO2 sticks to the charged amino acid residues on the globin chain instead of on the heme group. It has different binding affinities and binding properties. We'll learn more about hemoglobin in chapter 23, but fetal hemoglobin is a little bit different than adult hemoglobin, and that is for a very, very good reason. It's specific to the oxygen needs of the fetus and how the fetus gets its oxygen in the first place. So we have alpha and beta globins in our hemoglobin, and the heme group does the lion's share of binding affinity for oxygen, but there are other components that play a role, and one of those is the structure of the globin chain. So fetuses have alpha and gamma globin instead, and gamma globin has a little bit higher binding affinity with oxygen than the adult form of hemoglobin. So what that means is the entire tetramer of fetal hemoglobin is better at hanging on to oxygen and more grabby about oxygen than adult hemoglobin. If you think about a placenta is a shared structure between fetus and mother, and the blood vessels of the placenta basically are those of the fetus rubbing up near but not touching those of the mother. So that's how gas exchange between fetus and carrier of fetus is achieved. So if the binding affinity of fetal hemoglobin were equal to that of maternal hemoglobin, oxygen exchange between mother and infant would be inefficient. So the way that the fetus sucks up the oxygen more efficiently is by having that higher binding affinity hemoglobin. So that ends at birth. You see this big decline in the synthesis of gamma globin and this big increase in the synthesis of beta globin, and that's how you go from fetal hemoglobin to out-of-womb hemoglobin. So that shift with those declines and those increases happens in that critical first year of life. So it turns out hemoglobin is only safe to have intact when it is inside red blood cells. So you don't want free hemoglobin in your plasma. Why? Well, it's a large molecule, and what ends up happening is it will block your kidney tubules and cause renal failure if it gets a chance to. So we want to keep it in our red blood cells, and when it's time to dispose of it, we want to dispose of it in a series of ordered steps that do two things. One, mitigate any damage potential by keeping hemoglobin out of the plasma, and two, iron is a limiting nutrient, so we want to recycle that iron instead of peeing it out. So, worn out red blood cells are removed by macrophages, and this happens in the liver, the spleen, and the bone marrow. The primary sites are liver and spleen. The bone marrow is a bit ancillary. So if the spleen is removed during a splenectomy, we talked about this in my other class, spleen's a weird organ. It's a huge blood reservoir. Blood cells go there to die. But if you need it removed, you can totally get along just fine without it. The liver will take over. So macrophages in those organs are going to engulf and begin to destroy red blood cells. And then the heme is separated from the globin, and each of those components undergo different breakdown processes. So globin is broken down to amino acids. And of course, the amino acids are available for protein synthesis. So you've got these 4 chains of amino acids, and they are, of course, useful. So you would just basically take them down to the studs, so to speak, and use them to rebuild. The heme residue has the iron removed from it, and the iron is returned to the bone marrow. And then sometimes it also makes its way back to the liver because the liver is a site of iron storage. So, why is poop brown? Do you have children? Have they ever asked you this? It's a good question. If you didn't already know, now you can answer. You may have thought, well, it's brown because it's brown. It's poop, right? Wrong. It's because of blood. So inside of the macrophage, the heme group has the iron removed, and that step converts the heme to biliverdin, which instead of reflecting red, reflects green. Biliverdin is then converted into bilirubin, which reflects yellow light, and bilirubin is released into the blood. This is the reason for the slight yellow color of plasma. Hepatocytes are going to remove bilirubin from the blood and secrete it into the intestine. Intestinal bacteria, I want to say that this happens via bile primarily. More on that later. We'll talk about the biliary tract when we get to digestion. Intestinal bacteria, primarily those of your colon, convert bilirubin into urobilin and stercobilin. So, those two contribute to the brown color of feces. So this is the answer to why is poop brown. And then Mark has included this amusing analogy to Bed Bath & Beyond. I don't want to ruin Bed Bath & Beyond for you if you're one of those people that finds it to be a magical place of home restoration, but this is still funny. Alrighty, so here's a summary slide of recycling. So I'll just walk you through this real quick since I already did. I just like this one because it has pictures, and I know a lot of you are visual learners, so they like when I can point to things while I talk. So we've got new blood cells coming in response to from the kidney. Erythropoietin stimulates the production of new red blood cells. So those bop around for 120 days, and they circulate about 700 miles during that time. Once they're old, they either rupture in the bloodstream. Fortunately, that usually only happens very rarely because mass hemolysis is bad. So 90% of them get eaten up by macrophages whole, and this is in the liver, spleen, and bone marrow. So the iron gets removed from heme. The iron is transported back to the bone marrow by transferrin. Trans meaning transport, and ferrin meaning ferrous iron. And that is used to make more blood cells. And then if the bone marrow runs out of iron, it can ask for some from the liver. That's how that works. The macrophage is going to convert green biliverdin into yellow bilirubin. Yellow bilirubin is added to the bile along with bile salts. Bile is a fluid that helps you emulsify fat. So it's secreted into your intestines in order for the fat to be emulsified, and then bacteria in the colon are going to convert bilirubin into urobilin and stercobilin. Some urobilins end up via absorption with water back in the bloodstream and are eliminated in the urine. Others are eliminated in the feces. So this is why pee is yellow and poop is brown. Now, as you may know, because a lot of you are either already medical professionals in some capacity or are aspiring ones, the color of feces can tell you a lot about patient health, including about the state of their blood. So, for example, greenish feces can indicate issues with liver function due to the failure of bilirubin conversion. Feces that are dark black indicate hemolyzed blood in the stools, so those are indicative of a GI bleed somewhere. Usually a GI bleed that is higher up so that the blood has time to be processed and to clot. So there's all kinds of things that your feces color and consistency can tell you, and some of the things that it can tell you are things about your blood. So if you're curious about that, or if you're anticipating becoming a nurse, one of the things you'll have to become familiar with is the Bristol stool chart, which is a chart about poo and what it means. So I think that's enough time for one video. I'm going to get to work editing and posting the 2 that I've made today so far, and so hopefully those will be up on YouTube for you in very short order. I want to thank you again for your attention, and I will see you in the next video when we discuss fun things like, let's see what's next, shall we? Erythropoiesis. All right, have a good day, everyone, and I'll see you next time.